Thalassemia is an autosome linked recessive blood disorder. That means it is carried on an autosome and not a sex chromosome. And as you can see here, it is transmitted to the offspring when both the parents, both the partners, are unaffected by the disorder, but they are carrying the recessive allele, which can be transmitted to the offspring. So both have to be heterozygous for the progeny to have thalassemia. Now, to understand thalassemia, first you have to know what is anemia. So, anemia is when the red blood cells are not enough in the body because they are destroyed because of some deformity in the red blood cell. Okay, so red blood cells we all know have hemoglobin. So, this hemoglobin, as we have learned in the 11th standard already, it is made of more than one globin chains. Okay, so to be a little more precise, you have around two alpha chains and you have two beta globin chains. So thalassemia is of two types, alpha thalassemia and beta thalassemia, and therefore this what type of thalassemia would be dependent on what gene is getting mutated or deleted or affected. So if it is the gene that is coding for the alpha chains, it will cause alpha thalassemia. And if it is something that has gone wrong, or mutation or deletion with the gene that is coding for the beta chains, then it leads to beta thalassemia. So this has to be kept in mind. Okay, so alpha thalassemia, as I told you, will be caused because the alpha globin chains get affected, and beta thalassemia will get uh, or will be caused if the beta globin chains are affected. Either they are not synthesized enough or they are totally absent. So what are the genes that control this disease? So you have the HBA1 and the HBA2 genes, okay, which are primarily involved in alpha thalassemia, because these two genes, which are located on chromosome 16, they provide the instruction. These two genes provide the instruction to make up this protein alpha globin, which is required to make the alpha globin chains of the hemoglobin. So if these two genes, that is HBA1 and HbA2, are defective, then alpha globin synthesis gets affected. Now, please note, since genes are always in pairs, so you have HbA1, you will have two copies, and HbA2, you will have two copies. So, these are the genes that control alpha thalassemia. Coming to beta thalassemia, it is the HBB gene, which is located on chromosome 11. So, again, here HBB gene, if it is a normal HB gene on chromosome 11, it will happily make this protein called as beta globin, and the beta globin hemoglobin chain will be normal. But if there is something wrong, this gene gets mutated, deleted, gets faulty, then it will affect the beta globin chain of the hemoglobin. Okay, now red blood cells do not work properly in thalassemia. I told you it's a kind of anemia. The red blood cells are affected because ultimately hemoglobin is contained in the red blood cells. So the red blood cells, they stop working properly. The hemoglobin that is produced is faulty, and the patient needs to take a regular blood transfusion. So, throughout the person's life, he needs an influx of blood because his blood does not have enough of RBCs and he is constantly tired and anemic. Now, how does thalassemia differ from the other blood disorder that we have studied in our previous tutorials, that is sickle cell anemia? Let us see. So, both thalassemia and sickle cell anemia, I said, were forms of anemia where the red blood cells are affected. But the underlying difference is that thalassemia, as you can see marked in red here, is a quantitative problem where too few protein molecules will be synthesized because something has gone wrong with the gene. Whereas sickle cell anemia is a qualitative problem where it is not with numbers but it is incorrectly functioning globin proteins. Okay, so defective protein, not too less, but more the qualitative aspect of the protein gets bad. This is to be remembered. So, this is all in thalassemia. Now, you can just see a picture here where you can compare the normal blood components like the RBCs you can see here, and this the malformed red blood cells in thalassemia. You can see how they are malformed. So definitely these the oxygen carrying capacity of these blood cells will get affected. And these in white and cream color here are your white blood cells and your platelets. So basically, you can see that in thalassemia, a normal person's RBCs, the shape, and here you can see the shape of this. Okay, and how do they look in case of a person suffering from thalassemia?